webinar. So in the response to the SARS-CoV-2 pandemic, the use of mRNA and development of vaccines has been really crucial. The rapid sequencing of the virus has allowed us to have a better understanding of its genetic makeup, leading to the development of very highly effective vaccines. And real-time monitoring of the virus's evolution through next-generation sequencing techniques has allowed for the rapid adaptation of vaccines to new variants. And we will explore the impact of these advances on the current pandemic response, and then what it means for the future of vaccine development for this and other viruses. I think that we'll have a great opportunity to gain insights into the latest breakthroughs in genomics and vaccinology. Just to introduce myself, my name is Emma Thompson. I'm Professor of Clinical Infectious Diseases at the MRC University of Glasgow Centre for Virus Research. I'm joined by my co-host, Julian Knight, who's Professor of Genomic Medicine at the University of Oxford, who you probably mostly know well, and who is also an expert in genetic variation and how that can impact genes critical to mounting appropriate immune responses and susceptibility to infectious, inflammatory and autoimmune diseases. Now, moving on, I'm going to introduce our first speaker, Professor Sharon Peacock, CBE, who is a British microbiologist and is Professor of Public Health and Microbiology at the Department of Medicine, University of Cambridge. She's very well known for her use of microbial whole genome sequencing for bacterial infections, but in 2020 she became Executive Director and Chair of the COVID-19 Genomics Consortium, or COG UK. And she oversaw the development of a network that catapulted the UK to prominence as a leading authority on the generation and use of SARS-CoV-2 genomes as part of the pandemic response. The emergence of variants and the need to detect and track these represented a major shift in thinking and practice for disease control and vaccine effectiveness. And perhaps I can now welcome Sharon to give us an introductory talk. We will then move on to Tess Lamb, who I'll introduce before her talk. Sharon, if you'd like to.
Yeah, thank you so much, Emma, and thank you so much for inviting me to talk today. It's a great privilege and pleasure to be here today. Okay, so let me just put my slides up. The moment of truth. Can you see my slides okay? Brilliant, okay. Well, the title of my talk is Lessons Learned from Sequencing During the Pandemic. And I've split it down the middle. I'm going to talk about what we learned about the virus, but I'm also going to talk about how we learned to do rapid sequencing very well and for the benefit of the pandemic response. And so the lessons learned really are kind of, I suppose, a scientific lessons learned, but also a kind of a reflection on how we managed to stand up such a large consortium in such a short space of time and deliver a large number of genomes. Okay, so we were not the first person to sequence the virus, not by a long shot, actually. So there was a tweet by Eddie Holmes on the 11th of January 2020, saying that there was initial genome sequencing of coronavirus associated with the Wuhan outbreak. And that was then reported in full in January. And that information was absolutely key to vaccine development, because we then understood that this was a coronavirus, that it had a spike protein gene, etc. And you may well hear more about that from Tess, but that was the vital first clue, what we were dealing with here, what the cause of the infection was, and how one might respond to it in terms of vaccination development. But actually what the UK did do was it was amongst the first to develop a capability to generate SARS-CoV-2 genomes at scale and use this for public health, because in the past, we've tended to use sequencing of pathogens quite reactively and often used to look back to solve kind of a query or a question. What we did here was we used it to guide the pandemic response. And we really showed beyond any doubt, I think, that sequencing generates actionable information that supports an effective response through vaccine development and redevelopment, understanding transmission of the virus between people and across the world. And so I'm going to just talk a little bit now about the basis of what we were really looking for. So what we were really looking for was to track viral mutations. And so mutations in the genome are the basis of most evolutionary change. And RNA viruses have high mutation rates, actually, up to about 1 million times higher than the human genome. The issue is that changes in the virus have the potential to allow them to adapt to their environment and continue to infect humans. Now, this shows a schematic of the viral genome of SARS-CoV-2. It's around 29,000 nucleotides long. And you can see in the pink the gene that codes for the spike protein. Now, the spike is important because the spike is studded onto the outside of the virus, and that's where the word corona comes from. It forms a crown. And that spike is really key because it's like a lock and key. It's the thing that recognizes the human receptors or the way into our cells so that it can replicate. And some of the work that Emma's done on how the virus has got into cells and how that's changed over time has been a really elegant description of virology during the pandemic that really helped us to understand disease severity and other aspects. So this spike protein is really key, and it's the place where our immune response is targeted to, because if we can get an antibody blocking the spike, then we can prevent the virus from attaching and entering. So SARS-CoV-2 mutations, they happen probably about twice a month in the generation of a virus. And there are 3 outcomes. Most mutations that can occur, they're completely random. Most mutations, they don't affect the virus behavior at all. Some mutations will disadvantage the virus, actually, and though the progeny of that virus will not be observed in the population or will dwindle. What we really want to know about is the mutations that cause an advantage to the virus in terms of its fitness properties compared to everything else that's circulating. So this is a process of natural selection where the fittest will win out. Now, having trained as a clinician, and the ABC of resuscitation was drummed into me, airways, breathing and circulation, the way I think about variant assessment for COVID-19 is to think, is the virus more transmissible? Is it likely to evade the immune response, either because we have had natural infection before, we have antibodies, or we've had a vaccination? Or is this going to cause more or actually less severe disease? So this is the variant assessment. But however, spotting important variants was very difficult and still is, actually. So spotting which mutations are associated with the change in biological behavior. If we go back to September, October, November last year, we had very little understanding of what that actually meant. And over time, we've accumulated a catalogue of mutations that we believe are associated with particular characteristics. But initially, it was a very steep learning curve for us, and spotting important variants in amongst the many thousands of variants that were circulating was challenging. And this is a schema that was developed by public health agencies, including PHE and US CDC, where really what you're doing is you're horizon scanning for the behavior of the virus in the community. Is it reinfecting people who should be immune? Is it spreading more quickly than you would expect normally? So horizon scanning really is looking at the biology of the virus in the community. And if there's a signal, such as it seems to be spreading more, then it becomes a variant of interest. And at that point, the investigation really begins in earnest to watch what that variant is doing. Now, we know there's been a series of variants of concern, and these are ones that spread more rapidly and or evade our immune response. And then very fortunately, we haven't seen the top of that ladder, which is a variant of high consequence, which would be a virus, for example, that caused much more severe disease, completely avoided our immune response, etc. So this is one of my favorite pictures of all time, actually, and it's an image from MicroReact, developed in Oxford by David Anderson and his colleagues to look at the variants in the UK circulating over time. Now, if we look at these, this is a timeline from March 2020, and it actually ends quite shortly after Delta has really become dominant in July 2021. If you look at wave 1, and each color is a variant, not a variant of concern, just a variant, a specific combination of mutations in the virus. You'll see in wave 1, there was a huge number of variants that were circulating, and they were all, I guess, competing against each other. Nothing was really escaping. Then at the beginning of wave 2, we saw something called B1177 in the red, and it expanded to create, it was causing around 30 or 40% of all cases in the UK. And detailed investigation demonstrated actually that this wasn't due to the virus, it was due to holiday behavior. So people were going to Europe, they were acquiring this in Europe and introducing it into the country, and then it would spread. And so things that expand aren't always to do with the biological behavior of the virus. But when the Alpha variant emerged, which you'll start to see really starting to begin to emerge in around middle of November onwards, what we then realized was that this virus was spreading more than it should do, considering we had already gone into lockdown. And what you'll see here is this pattern of Alpha completely wiping the board clean, and it went on to sweep across the rest of the world. And then Delta came along and swept that away. And then, of course, there's been Omicron since. So what this again shows really is how we learned about the virus. We fully understood the rules that we were playing with in terms of, you know, Darwin's natural selection, but it was really only in December and onwards that we saw these sweeps of variants that were clearly fitter than other variants that were circulating. And of course, we know that Alpha variant emerged, was detected in December. And we can talk about this if you're interested, but it had acquired multiple mutations. There's been quite a lot of discussion about what appeared to be kind of supercharged evolution in this variant. And it was more transmissible, about 50% more transmissible. Now, we called this the Grinch variant because actually it did steal Christmas from everybody. We had to go back into lockdown, but COG UK were called back into work over Christmas to sequence as fast as we could to understand this virus better. And since then, we've had waves of variants. In fact, this doesn't even come up to the minute, but you'll see the way we've had sweeps of variants go through our population because they have an advantage in transmissibility and or vaccine escape. This is one of my favorite images of the relative fitness of SARS-CoV-2 variants over time. It looks quite complicated, but it isn't that different, isn't that complex. It was published in Science in 2022. And on the y-axis, you've got the fold increase in relative fitness of a variant compared to the original virus, which we call the Wuhan lineage. And what you'll then see on the x-axis is the date of lineage emergence. The size of the bubble is the number of people who were diagnosed. It's a cumulative case count, effectively. And what is extraordinary about this is that the Omicron is, as you can see at the top right-hand corner, much fitter than other variants, but fortunately causes less severe disease. And again, the work that Emma did was key to understanding some of the basis for that. So what's the future scenarios for the SARS-CoV-2 virus? Well, there are 4 possibilities. One is that the virus becomes extinct. It just dies out pretty much through minimal control, or the virus isn't really very fit. Global eradication through vaccination, for example, with smallpox, we achieved that. Elimination through zero COVID policy, well, we know that that was attempted, but didn't succeed in the end. And endemicity, that's where we are at the moment. We're going to live with this virus for the foreseeable future. So just as I come to a close, I'm actually going to think about my personal top 8 lessons. I don't know why 8, it's just I added them up this morning after having done the slides. And I want to run through how we can think as scientists, not just about the science, but the way we behave when something like this happens. Now, the first thing that actually helped us to respond very quickly to sequencing was how well prepared we were for the future. We had deep roots, so we had an academic engagement on pathogen genomics for well over a decade. We had equipment, consumables, know-how across the UK. And the 100,000 Genomes project was largely focused on human genomes announced by David Cameron, but we actually, as a recommendation from that, said that sequencing of TB, HIV, and hepatitis C should advance at pace for the benefit of patients. And so the expertise of applying genomics was really beginning to gather pace. We also had real-world experience and practice with outbreaks such as MERS, Ebola, and Zika, and the CMO was behind us in publishing a chapter in her volume 2 2016 Generation Genome, and that highlighted the really many benefits for sequencing of bacteria and viruses. Science investment was really important, so we had the MRC UKRI funded CLIMB, which is a cloud-based infrastructure for microbial bioinformatics. That's where all the analysis goes. And the methodology network for doing sequencing called Arctic was really thriving. And of course, we have the kind of stupendous Wellcome Sanger Institute, a mighty institution that can do very high volume sequencing. So unlike for other areas of our response, we were well prepared for this. We also acted at pace, and we were funded at pace. And so I'm not going to go into detail in this timeline, but it's a timeline of March 2020. And you can see on the top the number of genomes we had sequenced during March, and on the bottom, the number of cases that were diagnosed during March in red, and underneath the number of genomes we had generated. So what we decided to do was a group of scientists got together on the 11th of March at the Wellcome Trust to decide a blueprint for how we were going to be sequencing this virus. We wrote within a couple of days a proposal that we submitted to Patrick Vallance and Chris Whitty, and they agreed to fund that by the end of the month. But by the middle of March, we were already getting teams underway to think about how we collect samples, how we sequence, how we link the genomes to the metadata, how we report right, etc. And something called CLIMB COVID was created, which is our cloud infrastructure. So acting at pace was really important. Teamwork was also really crucial. So this consortium had 21 partners. The 4 public health agencies of the United Kingdom were really the kind of the levers for implementing what we found. And 16 academic institutions across the country. So you can look to see if your institution is there, and also the Wellcome Sanger Institute. And we had a consortium agreement that was a wraparound, a legal framework that allowed us to work together. But teamwork was key, as well as a focus on impact over process. Another lesson I really learned was I had to work in an imperfect system, and so did everybody else. So people said that it was like rebuilding the plane whilst trying to fly it, and we all had a piece of it. So we were desperately trying to get through the day, and the whole system was constantly changing. So a new lighthouse lab would open, for example, we'd have to get their samples or another diagnosis. There was lots of things changing the whole time. So I didn't find it particularly comfortable as a scientist who likes to be, think about kind of the perfections in life, but this was an imperfect system that we were working in. And it was also very chaotic at times. We also innovated, but we didn't have a central control and command. We devolved the innovation to teams, and we had devolved leadership. So we had different teams being led on logistics, methods development, data linkage, and analysis tools. And to devolve that was really important for us. We also shared what we created. So the work of COG members was adopted worldwide. We had sequencing methods, the Pango lineage, informatics tools, and we released all of our genome data immediately that we had it to the world. I think this is my 7th lesson. We got someone else to mark our homework at the end of the day because we wanted to know where we'd done well and where we had done less well. And so RAND Europe was asked to do a full evaluation of our performance. It's on the internet should you want to go and see that. And so important to finish with actually is that it's the people that really matter. And one of the things that we did was there were about 600 people in our consortium, mostly volunteers. Our money went on sequencing rather than on salaries. And so the people really made this work, but we shared the publication dividend by ensuring that we had a COG UK title that went into every paper that used our information. In that way, we could honour the people that have helped us make this consortium a spectacular success. So a big thank you to all of those people and their families, actually, particularly over Christmas 2020. So I'd like to thank you for listening, and it's been a pleasure to talk about this subject to you today. Thank you.
Thank you very much, Sharon, for such an inspiring talk. And we will have a chance to ask you questions shortly. But and can I just say to those listening in that if you do have questions, please post them on the Q&A button. The chat will not work, so please post on the Q&A, and we will come to your questions in due course. Thank you, Sharon, for the fantastic talk. We'll come back to you. And our next speaker is Professor Tess Lamb. Tess is Professor of Vaccinology and Immunology at the Jenner Institute at the University of Oxford, and is one of the co-developers of the Oxford AstraZeneca COVID-19 vaccine. Importantly, the Lamb Group are also currently developing and testing vaccines against a number of other potential outbreak pathogens, including Crimean Congo hemorrhagic fever virus, Lassa virus, Nipah, influenza, Ebola virus, and coronaviruses in general. A number of these vaccines have progressed to clinical trials and may be important for future emerging outbreaks. And with that, Tess, I wonder if you might like to share your slides. Thank you very much.
I shall try. Can you see the slides and hear me well?
Perfect.
Great, thank you, Emma. First of all, thank you to the organisers for inviting me here to speak. I'm going to talk about the lessons that I've learned from COVID-19 and the journey that I've had across the last 2 or 3 years. So a little bit of a disclaimer that my journey will be different to everybody else who's worked in the clinical trials. I want to take you back to the Ebola outbreak that started in 2013, 2014, and went on to claim an estimated 11,000 lives, an estimate that is largely thought to be underrepresentative of what actually happened. And after the Ebola outbreak in Western Africa, there was a number of task forces set up. The WHO and other government bodies generated a hit list, for want of a better word, of those pathogens that they thought might generate the next pandemic, epidemic, or outbreak. And there was a call to arms. There was a call put out to generate vaccines and therapeutics against all of these pathogens of concern. And some of these, I suspect, a large number of you will have heard of. Everybody's probably heard of Ebola virus and Zika, Crimean Congo hemorrhagic fever, which I think is a favorite virus of both myself and Emma's. Doesn't tend to roll off the tongue, but is very impactful. And Rift Valley fever as well is a little known virus. Regardless, we at the University of Oxford and many other places, including some pharma companies, started to make vaccines against these pathogens of concern. So I wanted to go into a little bit of detail, not too much, about how to make a vaccine, because it is relatively easy. It's not the witchcraft that some people seem to think it is. But first and foremost, you need to know a little bit about the pathogen that you're trying to make a vaccine against. And that's where genomic surveillance and sequencing is hugely important, so we can identify the pathogen that is causing the outbreak. And once you've identified the pathogen of interest, what you really want to do is identify a part of that pathogen that you think, if you have a strong immune response against it, you'll stop infection. So Sharon has already covered some of the details of the spike protein, and that was the most obvious target for the vaccine manufacturers to use in making a vaccine, because the spike protein is how the virus gets into your body. And if you can neutralize that threat, then you're going to be able to protect your vaccinees, your volunteers in your clinical trials. And two platform technologies, vaccine platform technologies that really came to the fore during SARS-CoV-2 pandemic were viral vectored vaccines and RNA technologies. And in essence, they both work in a similar manner. They both take the recipe or the coding sequence, if you will, for the spike protein, and they get it into your body. And once these platform technologies has carried the recipe for the spike protein into your body starts to make large amounts of spike protein. Your immune system recognizes it as foreign or alien and generates both antibodies and T cells. And hopefully, to a level that you will be protected when you see the actual pathogen, when you come across SARS-CoV-2. And really, these platform technologies were seen as the darlings of the COVID pandemic. They were the ones that were hailed as revolutionary, as the next best thing. But I really think one of the lessons we need to take away from the pandemic is not to de-invest in the other technologies. Whole inactivated vaccines and protein subunit vaccines have a huge part to play in preventing infections routinely, and are used routinely in our healthcare system within the UK. So while viral vector vaccines and RNA technology have got a lot of attention across the last 2 or 3 years, one lesson that I have learned is that we can't de-invest in all of these techs because we don't know what is coming next, and we don't know which technology may work better. And truth be told, viral vectors have been around, and RNA, for decades. In fact, the first viral vectors that were licensed were vaccines that were licensed against Ebola virus. And they were trialed and tested in the outbreak that happened in 2013 to 2016. A question that we frequently get when we're giving talks is, how were we able to go so quickly when it normally takes you 5 to 10 years to generate a therapeutic or a vaccine? And to speak to that, I thought I would show you what the normal development process and timeline looked like before COVID. And this was a linear process where you started with designing your vaccine that would generally take weeks to months, depending on how complex your pathogen was. It can be sped up. Myself and the other co-designer designed the Oxford AstraZeneca over a weekend, so you can do faster. The next step is to start doing preclinical studies, and you normally start with small animal studies here, and you're looking to see if the immune response is such that it is the type of immune response that you would want. So typically neutralizing antibodies and a good T-cell response. And if you don't get that response, then you go back to the drawing board, you start designing your vaccine again. Because really, as you progress along this timeline, each of these steps are not a small increase in price or time or investment, they're astronomical. So we're quite lucky in Oxford in that we have a we have a GMP manufacturing site where we can make vaccines that can go into clinical trial. But even so, those vaccines will generally cost between $1 million to 1.5 to generate. And by the time you've got to phase 3 GMP or onward licensure, you really want to make sure that you've got the best in class product, that you're progressing what you think is a game changer, a market changer. And to do so, you need to invest a lot of time, a lot of effort, and a lot of money. And that is typically why vaccines or therapeutics take 5 to 10 years to develop. We looked at this at the start of the COVID pandemic, and we realized we couldn't go that slowly. So how did we actually go more quickly? Well, we took a risk, and the risk we took was purely financial. And it wasn't a risk that I took, it was the UK government's money, it was Serum Institute, AstraZeneca, and other investors that allowed us to start our phase 2 and 3 clinical trials while we were still running our phase 1 clinical trials. And I hasten to add that while I say we did work at risk, we always prioritize the safety of our volunteers. I suspect speaking to this audience that that goes without saying, but it doesn't help reinforcing that point because we frequently get asked that question. So another lesson that I've taken away from the pandemic is if you're going to go quickly, you need some plans in place, and you need to plan for the future, and you need to plan many steps ahead because things will, can, and do go wrong. You absolutely, to my mind, need to partner with pharma early, and you need collaboration. Without that open collaboration between government regs and international bodies, there is no way that there could have been a vaccine in less than a year. So those silos, those walls came down between all of those key stakeholders, and we were all pulling in the same direction. So to my mind, that was one of the most important outcomes, is collaboration and an open dialogue that allowed us to make a vaccine in less than a year. So what were the other types of learnings that I've taken away from running these clinical trials? Well, to set the scene, we made our first batch of GMP. Our first doses were made and filled in early April. The vaccine was released to trial towards the end of April. And at that point, we had a number of very lovely volunteers who even came to clinic without sleeves, so they didn't have to roll them up, ready and able to get vaccinated in their arms. We partnered with pharma, again, relatively early on the 30th of April. And what had started to happen across those early months really took off when we started our clinical trials. And that was the fake news and misrepresentation and downright lies in some cases around what was happening in these trials. So our lovely volunteers who came forward and were vaccinated woke up to fake news that they had ceased to exist 3 or 4 days after being vaccinated. And one of the tough lessons to learn was no matter how much you try and do to dissuade or get that genie back into the bottle, get that fake news disproven, it doesn't matter. Once it's out there in the ether, there will be people who believe it, there will be people who will propagate it. So we really need to tackle this head on, and we probably need to do it in a more proactive way than we necessarily did during the pandemic. I have to admit that it was a steep learning curve for me and many of the other team members in communication and how to get out front and in front of these stories. The other point that I would like to make is around the efficacy readouts that came towards the end of 2020. And sometimes science is difficult, science is hard, it's not easy to explain, and certainly our efficacy readouts were not easy to explain. But if we don't do it well enough the first time, then we should go again and we should explain it so that people can understand. And I'll take this opportunity to run you through some of the confusion around why we had 3 numbers and everybody else had come out with just 1 number. So within the UK, we had 2 different vaccination regimes. We had individuals who had had a low dose and then a standard dose. And we also had individuals within the UK and in Brazil who had received 2 standard doses of our vaccine 4 weeks apart. And then efficacy readout that was associated with those individuals that had 2 standard doses was in and around 60 to 64%. And for the low dose, followed by the standard dose, it was much higher at 90%. At the time, we had done this analysis over a weekend, and it had been independently verified. And when you are working with a large pharmaceutical, there is an onus on you to come out with these results as soon as you know them, because they can impact share price. We weren't hiding data. We didn't know why there was a difference between those regimes. We were able to demonstrate after a couple of weeks or some time that the reason there was a difference between the efficacy associated between those 2 regimes was largely down to the interval between the doses. So in those individuals who had had a low dose followed by a standard dose, there was a longer interval. And that longer interval was associated with a higher antibody titer and a higher efficacy readout. So really, again, communication was critical. And would I go back and try and simplify the message and give 1 number instead of 3? No. I think sometimes we need to be driven by the science, and science is hard, and we need to explain it. The one thing that I would suggest that I would ask the audience, dissuade them from doing, is comparing across clinical trials. Everybody does it, including myself, but it's wrong. Generally, clinical trials are set up for a snap period of time or in a certain group of people, a defined population with a defined variant, and also the primary outcomes and how we define disease versus other manufacturers are different. So really comparing across trials, you're not comparing like with like, and you're doing the data a disservice. We were really encouraged when these vaccines went out into the wild, and they did what they should have been doing, protecting our most elderly, our most frail, and making an impact against COVID in older adults and the most frail individuals. And this certainly was a lesson that I learned, looking to see real world data to back up your clinical trials. It was really impactful and something that I still remember to this day. The other thing that I've learned is that I need to become, as we all do, better communicators. The importance of communication was really highlighted around the level of misinformation that got out there. The anti-vax movement that really tried to dissuade individuals from having vaccines. And anti-vax is very different to vaccine hesitant and vaccine reluctant individuals. Vaccine hesitancy, in my experience, generally stems from people who have questions. They don't quite understand something, they're curious. Having an open and honest dialogue with those individuals can help them decide whether they will or won't have a vaccine. They're generally very reasonable individuals who just don't know what to do. The anti-vax movement is something different. It's a different beast. However, anti-vax sentiment is nothing new. This is a cartoon from the time of Edward Jenner. Edward Jenner is frequently referred to as the grandfather of vaccination, although there were many people doing variations of vaccination before and at the same time of Edward Jenner. Regardless, he noticed that milkmaids who had been exposed to cowpox did not go on to develop smallpox, a devastating disease claiming an estimated 300 million lives. He performed some what we would consider relatively crude clinical trials in the late 1700s and 1800s, and was able to demonstrate that if you were exposed to cowpox, you would be protected against smallpox. And this was a cartoon at that time suggesting that if you were to get Edward Jenner's vaccine, you would go on to develop bovine-like characteristics. Nobody has turned into a cow because of vaccination, but this type of sentiment, this type of message was not dissimilar to the type of messaging that was being propagated around the Oxford AstraZeneca vaccine and it being called a monkey vaccine. Having said all of this negative messaging, I do want to highlight that we received many, many words of thanks, many very generous gifts, one being very generous brownie delivery. So for every bad message we got, there were lovely members of the community and the public who did reach out to us to thank us and support us. But fundamentally, I don't know that we're doing a different, we're approaching the anti-vax movement in the right manner. I don't know that we're doing a better job at actually tackling misinformation and doing it in such a way that we can help individuals who are hesitant or reluctant make a decision in the best informed manner that they could be. The other message or the other take-home point that was very evident to me and many others during COVID was the slow rollout of vaccines to lower middle income and low income countries, and how when the vaccines started to be generated, that most, if not all of them, were going to upper middle income and high income countries. That needs to change for the next pandemic. Certainly the Oxford AstraZeneca vaccine, the ethos was to generate a vaccine for the world. This is old data, but something that the team are hugely proud of, especially that 2/3 of our doses went to low and lower middle income countries, and that in excess of 3 billion doses were released to supply. But we really do need to tackle this disparity in where the vaccines went better, because without us all being protected, no one will be protected. So to end in a similar note to Sharon, what were the lessons that I've taken away from concept delivery of these clinical trials? But when a rapid response is required, it absolutely necessitates creative approaches to funding and research. And for that to be properly enabled, we need collaboration across government regs, charity, industry, and academia, and we need an open and frank dialogue. We absolutely need local and international capacity, and we need that to be a sustainable model. We need international capacity to run trials, to manufacture vaccines, to roll out those vaccines, and a strong scientific base in those countries that were seen to be left behind during the SARS-CoV-2 pandemic, so that doesn't happen again. How do I think we'll achieve that? Much to what Sharon was saying, we need to invest in people and places. We need an open dialogue, we need sustainable funding, and we need in-country investment to really get the capacity built up and maintained. And in that way, I think we will be in the best position going forward for the next pandemic. It's only when it happens, not if. And in that way, we will be proactive and not reactive. And with that, I don't have a slide with everybody's names, unlike Sharon, because I will forget somebody. So I take the easy way out and just put a general icon up for everybody. And thank you very much for listening. I'm very happy to take your questions.
Thanks very much, Tess. That was a brilliant talk and really interesting and insightful, of course. So we're now going to move on to some Q&A, and just to remind the audience to put their questions in the Q&A so that we can make sure to ask them. Remember, don't try to put them in the chat, put them in the Q&A, and we'll pick them up over the next 15 minutes or so. And Julian and I are going to coordinate, I think, to ask some questions of you both. Maybe I can just kick off a little bit with a kind of general question. So both of you really have been at the center of innovation, both like in terms of the science, but also of models of working, particularly with academic centres, really hand in hand with industry and the NHS and public health. And I wonder what your opinion is more locally within the UK, how we should plan for the future to get the infrastructure right, to make our response easier for the next virus. And I'm asking that of both of you, so either of you jump in when you want to.
Do you want to go first, Tess, or shall I have a go?
No, please go ahead.
Yeah, okay. Yeah. Well, models of working. During times of the pandemic, it was really key that everybody pulled together, and Tess talked about silos. And there are silos between different types of organization, and each type of organization has their own particular culture. And so academic institutions are different between each other, and they're different from the public health agencies. And during the pandemic, it was clear that everybody wanted the same goal. And so what I saw actually was that there was no barrier. Everybody piled in and did what they needed to, and just worked at pace to get it done. I think the question you're asking, though, Emma, is, how do we make that stick rather than let it ebb away? And at the moment, for example, COG UK will end its funding at the end of March. And so there will no longer be a consortium for sequencing of pathogens across the country. And my sense is that that actually the importance of infectious diseases to the security and health of this country has been realised in full. And I think that we have narrowly escaped what's happened with other pandemic viruses. And I think that now it's clear that we have to invest, and we have to get those really solid foundations in, which I don't think we were, I don't think there was that level of awareness of what might come. So the first thing to say is public health agencies are absolutely key to having strength in our country to respond, because they are the first responders after all. If you have a fire, the fireman comes along. If you have an outbreak, the public health agencies come along. But in addition to that, we must make science really powerful, because we are the critical partners, critical friends that go along and they do the innovation with our public health agencies. So not only do we have to invest in our public health agencies and their capabilities, which I believe that they are the first responders, we have to invest in science. And for me, I have to say that initiatives such as Horizon Europe and such funding initiatives, there's been 2 big reports coming out actually in the last week, one from the new Department of Science and the other from Sir Paul Nurse. And we clearly need to invest in our academic community. So that's quite a long way of saying that I think that to prepare, we can't just recapitulate what we did before, which was done in an emergency, but preparation means training people, investment in public health agencies and in our academic community. That was quite long, wasn't it? I'm sorry about that, but
it's great. Thanks, Sharon. I guess we have new tools, right? We've got new tools that we can use, and they have to be placed in the middle of what we do next time as well. And then there will be more tools then, hopefully, also. Tess, would you like to also add something to what Sharon said?
Sharon's covered it really well, so I'm quite happy she talked for a while. I agree, Emma, that there will be new tools, there will be new innovations, but you're not going to have those new tools or new innovations if we don't invest in them. I don't want to do what we did again. I suspect Sharon doesn't want to do what she did again. So it's around future proofing so that the next generation or our children or our grandchildren don't have to have such a reactive response towards a pandemic. And I really do believe that investment now will safeguard us in the future. It's a bit like house insurance. You don't appreciate it until your house is burnt down. And I don't know necessarily that there's enough foresight that how much this investment will pay off in dividends going forward. And we do absolutely need to invest in the NHS as well, and our people who are tired and worn out, and make sure that they're in a better place next time around.
For sure, and afterwards. Gillian, would you like to ask something?
Thank you, and thank you both, Sharon and Tess, for wonderful talks. I was wondering if you could just expand a little on whether you've been surprised by this virus, whether it's behaved in the way that you might have expected, and indeed, you know, how different it is in terms of other similar viruses.
Shall I go first, because I'll be quicker?
Okay. No, I can do short answers, honestly.
No, I don't mean it that way. I'm not a virologist, is all I mean. So yes, I was surprised, Julian. I had my money on that this was going to be an influenza virus, that this was a wet market in China. It was going to be a flu virus. The next pandemic would be a flu virus. I was wrong. I was also very concerned because respiratory pathogens do impact children quite significantly, and that's not to play down the very real impact this virus has had on children, but it hasn't had the mortality rates that you're seeing in older adults. However, having said all of that, the way that the virus has evolved over time hasn't necessarily surprised me, but I will hand over to Sharon to talk more on that.
Well, thank you. And if I struggle, Emma, you're my next port of call. I would say that well, the first thing to say is that when the virus emerged, it was a fit virus. It was already perfectly capable of spreading. So it had this kind of R0, where you look at how many people one infected person infects. It's like 2.8. So one person will infect 3 people. And it had a couple of other tricks up its sleeve. First of all, the virus started to shed before you develop symptoms in people who are going to develop symptoms, so about a day before. So you're mixing with everybody, you don't realize you're sick, you're spreading it. The other thing is, of course, that we learnt, the lesson of asymptomatic infection. So people going about their business, they didn't know they were infected. So this was a fit virus, however you look at it. And in many ways, its behavior was completely predictable. I mean, it sort of played using the Darwin playbook, if you like, of natural selection. I think the really difficult point is when that was going to happen and really understanding what would happen. But presumably, as we developed our immunity through being infected naturally, the population would actually build up a level of immunity, so the virus would actually be selected for to transmit more readily than the previous version, so it could like outrun its competitors. And that's what happened. But actually understanding when that was going to happen, there was no playbook for that at all. And the difficulty is watching the variants to see whether there was one that would behave like that. The other thing I would say is that, well, there's probably two more. The other thing I would say is that when Alpha emerged, it was a bit of a shocker because it had lots of mutations all in one virus, and it was really quite unusual. And that kind of like supercharged evolution, I don't think people had really recognized that. And that was a bit of a shock. And that's likely to have probably the most plausible hypothesis is that it emerged after having been in somebody and who couldn't shake the virus off. I think that I think that the third thing that really there's been a lot of talk about really is whether the virus will evolve to become more or less pathogenic and cause more or less symptoms. And I think that Omicron we're in a good position because it's actually become less severe in its disease severity, and one imagines it might continue along that track, although we don't know for sure. But there's nothing that's evolutionarily kind of an advantage to the virus become a more severe disease, because you're likely to go to bed, not likely to mix. So there were some things that were predictable. The timing wasn't known, but there were some things that really did surprise me quite a bit, particularly kind of the degree, and Omicron too was hugely divergent. And so yeah, it was and finally to say that at the beginning we were only learning by watching and understanding what was happening, and the power of hindsight is incredibly powerful now, because I understood we understood what was happening, but at the time we were learning as we went along. And that's a really important lesson for me.
Fantastic, thank you, Sharon.
I was going to say that like Omicron, if you look at the way we responded to Alpha and the way we respond to Omicron also was quite a big shock, right? Because it had a huge number of mutations. And I personally remember being really alarmed by those mutations because we actually recognized many of them. And so it was a bit unlike Alpha, where we didn't understand what those mutations exactly meant at the time because we hadn't tested them in the laboratory. We hadn't linked them at first with sort of clinical data. But then Omicron again took us by surprise because actually it seemed to be milder than the Delta variant, which preceded it. And I guess perhaps that highlights the need for other, and not just genomics, which is incredibly powerful and helpful, and we can't have vaccines if we don't know the sequence and etc. We can, but it's better to know the sequence. But perhaps there's also a role for some of the more basic virology and making sure that the academic institutions are ready, and that it's almost written into the contracts of people who work within them that they should turn other things off and respond. I guess you're going to agree with that, but I remember being taken by surprise by that quite significantly. Can I pick up one other thing though, and just see what you think? Because one of the, like you mentioned, one of the critical groups of people who are seem to be at the heart of perhaps developing virus infections which are very evolved. How do we, where do we take that? We think perhaps that Omicron and other variants have evolved in people who have long-term or chronic infection. What should we be doing now to investigate that further, and also to evaluate how that affects those people, many of whom have got quite weak immune systems? Either of you could comment on that.
I'll give a brief answer because I'm just looking at the clock. My sense is that study of COVID-19 in the immunocompromised, who are the people we're trying to protect, is relatively understudied in many ways. And I think that they are the people we're trying to protect, and they are the people where variants could emerge. Although it is also hypothesized that people who are immunocompetent could also lead to a variant that's actually highly changed. But what do we do next? Well, for me, at the moment, it looks as if Omicron is sort of backed itself into a kind of an evolutionary corner. It's very transmissible, it avoids the immune system a bit, but it doesn't cause very severe disease. And there's nothing else on the horizon at all in terms of, it's just like offshoots of Omicron, which is like the kind of latest thing called Kraken or XBB 1.5. And that's more transmissible, but that's as far as it's got. And for me, I wouldn't be so bold as to know what's coming next. I would say that there's a reasonable chance that something else will emerge after Omicron, which is like an Omicron-like event. There's something really quite different that comes along. But I don't know what it's going to be, because we haven't been good at predicting what's coming and what it's going to look like. So all we can do is revert to watching, and actually watching very careful, particularly, if we haven't got the same capacity of sequencing, really selecting the populations who we study. So genomic surveillance and the role of the public health agencies, right back to, the importance of doing that.
That's great. Maybe we can just fit in one question from the Q&A. So it sounds like in the question, a need for a pandemic preparedness organization. The question is, what stakeholders would need to be included, and how do we get long-term funding prioritized in the UK?
I'll take the first half at least, because I'm part of PSI, Pandemic Sciences Institute within Oxford, but to my mind, and also Oxford Vaccine Group, to my mind, it needs a collaboration, a global collaboration. We can't be so UK-centric going forward. We need to have the type of interactions, collaborations that we saw during the pandemic, at the height of the pandemic, because without us running trials in Brazil, we would never have known the efficacy, et cetera, et cetera. Without the sequencing in South Africa and other places, again, we really need that kind of global collaboration. As to how to get long-term funding, if somebody could tell me how to do that, I'd gladly try it. That's slightly flippant. I would suggest that at the moment we invest, and we being high-income countries, invest a lot of money in security and safeguarding our borders against possible attacks. And perhaps for a small fraction of that budget, we might be able to safeguard our futures against the next pandemic.
Thanks very much, Tess. Was there anything you wanted to add to that, Sharon?
I don't think so, except that we have to obviously fund critical capabilities such as vaccines, diagnostic tests, genomics, virology, and so on. But we also mustn't be too blinkered, because actually funding the best science wherever it is, you never can predict what the next science breakthrough is going to be. And you often, so it's important that there's a broad portfolio of excellent science, and that does blue sky science to try and work out what the next breakthroughs are going to be, and not just trying to best guess that it's going to be, the 5 things you've worked out were important in the last pandemic. So breadth of funding and adequate funding to train the next generation of scientists.
Okay, well, with that, I think those are the last words, Sharon. Thank you very much. And thank you both very, very much for participating in this webinar. And I really would like to express my sincerest appreciation for that really interesting conversation, and for taking the time to share your expertise with us. I hope everyone enjoyed the session, and thank you to everyone for joining, and thank you. Have a nice evening.
Thank you. Bye-bye.
Bye. Thank you.
Bye-bye.